Welcome everybody. Okay, the recording is in progress. Thank you for this seminar for Escalon, and it's good to see a lot of familiar faces in here, good friends and good coworkers. And we, for those of you that are part of the General Assembly, we have the first hour, we have speakers, and then the second hour, we have a bit of a talk in the General Assembly. Today, we have two great speakers. We have Maria Belén Pisano and Dr. Xavier Velo. And I will start with Dr. Pisano. Maria Belén, I think you're first in the presentation here. Dr. Pisano is a researcher at CONICET in Argentina, and also the Universidad Nacional de Córdoba in the Department of Virology. She has done a tremendous job in hepatitis B and E, defining a lot of the molecular characterization in Argentina. And her talk today will be molecular epidemiology of hepatitis B in Argentina, genotypes, subtypes, and mutants of local circulation. And Belén, I think if you want to take the stage, do 25 minutes, 30 minutes, we do some questions, and then we'll go with Xavier.
Great.
Thank you very much.
Hello, everybody. Thank you, Jose. I'm going to share the screen. Let's see. Can you see?
Yes, we can. At least I can.
Great. Let me see. Okay. As Jose said, I'm going to talk about molecular epidemiology of hepatitis B in Argentina, about genotypes and subgenotypes and mutants of local circulation. I'm going to just to tell you genotypes that circulate in all Argentina, and then I'm going to focus on a work that we perform in Córdoba province, in the province where I work. So just as an introduction, the hepatitis B virus belongs to the family Hepatnaviridae, to the genus Hepatnavirus. It is an enveloped virus. The incubation period of this virus is about 30 and 150 days. It can produce acute hepatitis in the 90% of the cases when the infections are acquired in adulthood. And this virus can produce also chronic hepatitis in the 10% of the cases. Regarding chronic hepatitis, these cases can develop to cirrhosis and hepatocellular carcinoma. So the hepatitis B virus genome consists of a partial double-stranded DNA molecule of about 3.2 kilobases, which encodes 4 open reading frames, partially overlapped. Here is the representation of that. According to the genetic variability, this virus can be divided into 10 genotypes and many subtypes. The genotypes are defined with a divergence of at least 8% in the complete genome sequence. And the subtypes within each genotype are defined with a divergence of at least 4%. So the genotypes have a worldwide geographical distribution and are named from the A to J. So we can find genotype A mainly in Europe, also in Africa and America. Genotype B and C are mostly distributed in Asia, genotype E in Africa, genotype F and H are from America, and then we have genotype I and J from Asia too. Okay, so there is evidence that genetic variability can be associated with some clinical implications, such as different transmission routes, resistant to treatment, and progression to development of hepatocellular carcinoma. And there are current consensus that refer to this association between genotypes and clinical implications. Some examples are the European Association for the Study of the Liver and Argentine Consensus on hepatitis B treatment. These guidelines refer to the genotypes and the different treatments and clinical implications. So in the literature, there are some reports about this relationship between genotypes and clinical implications, such as modes of transmission. Genotypes B and C are related to perinatal and vertical transmission, while the other genotypes, horizontal transmission. Regarding tendency of chronicity, genotype C has a higher tendency, genotype C and A, sorry, have a higher tendency to chronicity. Genotype C and D has less seroclearance for the S antigen, the surface antigen. Regarding cirrhosis and hepatocellular carcinoma, genotypes C and D have been described to have a worse clinical outcome. Genotype C has higher serum levels of DNA compared to the other genotypes. And there are some mutations which have been described only in particular genotypes. And we have to say that regarding genotypes from the E to J, there are very few information in the literature. Regarding mutations, numerous mutations have been described in the hepatitis B virus genome, which with different clinical implications too. So there are descriptions of mutations in the ORF-S, which encodes protein S, which are related to failure in the diagnostic and immune escape. There are some mutations in the core and pre-core mutations too, which are mainly associated with a decrease E antigen production. There are mutations in the polymerase gene in the ORF-P, which are related to resistance to antiviral therapy. These mutations also are implicated in changes in the surface antigen because they have the open reading frames which are overlapped. So, well, what's the scenario in Argentina? Argentina, as you know, is a South American country. Here in Argentina, we have a low endemicity, less than the 2% of incidence. There are higher number of cases in urban centers in the city, and the vaccination is mandatory at birth, the first shot, and then at 2 months and at 6 months since the year 2000 here in Argentina. From the year 2012, the government invited us to vaccinate all the population. So nowadays we have universal vaccination against hepatitis B. Regarding genotypes, in the northwest of Argentina, we have mainly circulation of genotype F, more than the 90% of the circulating genotypes. In Buenos Aires, we have similar proportions for genotypes A, G, and F, but the last reports inform of high prevalence of genotype F, more than 50%. In the east of the country, in Misiones province, we have a lot of genotype D. This is related to the immigration process because Misiones has a lot of immigration from the east of Europe. In the Mar del Plata city, we have also almost 70% of genotype F. And in Córdoba, I'm going to talk about the genotypes that circulate here in our province. Córdoba is a central province in Argentina. And we aim, in our study, we aim to detect hepatitis B genotypes and look for relevant mutations. So we studied samples from patients with acute and chronic hepatitis B infection between 2010 and 2015. We studied acute hepatitis B infection. We studied 79 individuals, 68 of these individuals with mono infection with hepatitis B, and 11 individuals were co-infected with HIV. And regarding chronic hepatitis B infection, we studied 138 individuals, 82 were mono-infected patients, and 66 were co-infected patients with HIV. We performed PCR to these samples, PCR targeting two genomic regions, to the gene S and the precore basal core promoter region, which are here, and the S gene. Then we performed genetic phylogenetic analysis, and we investigated the mutations. So, well, regarding acute samples, we studied, as I said, mono-infected patients and co-infected patients. We found the main genotype were subgenotype F1b in mono-infected patients as well as in co-infected individuals. Then we had also a genotype A2 in both groups and genotype F4, subgenotype F4 for both groups. Here is a phylogenetic tree with the same results. Most of the samples grouped with genotype F was the genotype most frequently found. And regarding mutations of acute patients, we have we detected mutations in the ORF precore and basal core promoter for mono-infected as well as co-infected individuals. Most of the mutations were associated with a decrease in the expression of E antigen. And mostly we found these mutations mostly in genotype F1b. Regarding mutations in the ORF-S, we found in both groups, and most of the mutations were previously associated to immune escape and diagnostic failure. We also detected one sample with mutations related to resistance to the treatment in the polymerase gene. Regarding chronic patients mono-infected with hepatitis B, we found a lot of most of the patients infected with subgenotype F1b, followed by F4 and A2, very similar to the acute patients. We found in this group subgenotype F6, which we didn't have found in the acute group. This subgenotype is an Argentine subgenotype, which was first described in our country. Regarding genotypes in the co-infected patients with chronic infection, we also found in the first place subgenotype F1b, followed by A2 and F4. And this is the phylogenetic tree of all the chronic hepatitis B patients. We can see mostly the more frequent genotype, the genotype F, followed by genotype A, very similar to the acute hepatitis B patients. And this is a map showing our province. Here we can see the genotype distribution, geographical distribution in the acute infections and in chronic infections. And we can see that the genotypes are very similar, and the geographical distribution is also very similar. For chronic patients, we can find genotype F6. in acute patients, we do not see a subgenotype F6. Okay, regarding mutations for chronic patients, mono-infected patients, we also found mutations in the core, pre-core and basal core promoter, also related mutations associated with a decrease in the expression of E antigen and progression towards cirrhosis. This is all information which is described in the literature. And we also found mutations in the ORF-S, which again, mutations previously associated to immune escape and diagnostic failure. Regarding co-infected patients with chronic hepatitis B, we also found mutations for the ORF core and ORF-S, and we found some patients with mutations resistant to the treatment. So, well, as conclusions, we can say that genotype F was the most prevalent, followed by genotype A, particularly the subgenotype F1b. In all the studied group, mono-infected, co-infected, and with acute and chronic infections. Subgenotype F1b was found in a high prevalence. Subgenotype F6 was only found in chronic patients. This subgenotype has been recently described in Argentina. Detection of mutations in all groups of patients were performed, but with higher prevalence of mutations in chronic mono-infected individuals. This is very important in acute patients, mutations related to resistance to the therapy, because the physician can see which is the best option to do the treatment in case the patient needs treatment. We can find a low prevalence of mutations in the ORF-P in acute patients, and higher prevalence in chronic patients related to resistance to therapies. That's what I have just said. And we will continue with these investigations regarding genotype F, focus on genotype F, which is the local genotype here in Argentina. And we are going to start analyzing all the samples of Escalon project. Okay, thank you. This work was in collaboration with between Instituto de Virología here in Córdoba, in the School of Medical Science from National University of Córdoba, and in collaboration with the Health Ministry of the province, the government of the province of Córdoba. And we are thankful to Escalon project. We are going to start working with these samples. Thank you very much.
Thank you, Belén, for that great presentation. Good. So the floor is open for questions. I would like to start with a quick question. I have a few more, but I want to give the opportunity to others to ask. So you were talking about the F genotype, very prevalent in Argentina, and some of the subgenotypes in there. Do you have any idea, or can you say anything, whether that genotype has a stronger or weaker association with hepatocellular carcinoma related to hepatitis B? And I don't know if there's any evidence of that or not.
There is only one work in which associate more severe or cirrhosis, not hepatocellular carcinoma, but cirrhosis with genotype F. But there are preliminary studies and with a very little number of samples, so we don't know. That's the answer.
Okay, and then when you study mutations, you study them in the virus only, right? You did not have the chance to study kind of like mutations in the overall genome of the patient.
No, we have sequenced some. We have sequenced some samples, the whole genome sequence, but we don't have analyzed this data until now. But we aim to sequence the whole genome. Yes, it would be very interesting to look for other mutations.
All right. Anybody else has a question?
I would like to congratulate Belen for her work, which is really interesting. And I would like to comment of my surprise seeing that the genotypes more common in Cordoba are different than those which are the commonest in Porto Alegre, which is the capital city of a state in Brazil, which is quite close to Argentina. Around 10 years ago, in our university, we have performed a similar but probably more simple study than this you're conducting now, and we had maybe 35% of genotype A, 60% of genotype B, and only 5% of genotype F. So I was curious about this differences. Of course, there are different immigrations to one country and the other, but as we are geographically close and actually we share some similar immigration patterns, especially the Italians, I would maybe expect a more similar distribution of genotypes. But congratulations on your work.
Thank you. Yes, it is not surprising that the genotype distribution are different within regions. Even within a country, in Argentina, we have very different genotype distribution in the north, in the center, and in the south or east. And we think this has to do with immigration and with movement of people and immigration process. Yes. Buenos Aires, which is a cosmopolitan city, has a lot of genotypes, and in the north, for example, in which we have indigenous population, we have a lot of genotype F. So it is possible, yes. Thank you.
Any other questions? Marco.
Hi to everyone, and yes, also I would like to thank you and congratulate Belen for the work she presented.
Thank you.
I came a little bit late, and I'm not completely aware of the current literature on HBV genotypes, and I would like to ask you if which are the clinical implications of the different subtypes of F genotype. I'm asking that because in Chile we have only F genotype, but as far as I know, nobody have assessed the different subtypes, and I wonder which are the difference in terms of pathogenic mechanism and clinical implications in terms of developing of chronic liver disease or even HCC.
Yes, it's a very interesting question. We don't know. We don't know, and that would be very interesting. We only found different subgenotype distribution regarding chronic or acute infections. For example, with subgenotype F6, we only found subtype F6 in chronic patients, but we don't know. It would be very interesting to do some work about that. Yes.
And Belen, following Marco's question a little bit on the same line, I was curious about the subgenotype that you only found in chronic versus acute.
Yes.
Because I'm assuming that hopefully I can state this in an understandable way. So when you're measuring acute hepatitis B, you are a little bit biased, right? Because you're looking at symptomatic hepatitis B, which is the minority of the infections. So I wonder if that subgenotype that you find or not find there might have an implication in having clinical versus subclinical hepatitis B.
Yes, that's true. That's true. Some acute patients could develop chronic infections. So yes, that's true. It's just a descriptive study, but yes, you're right.
And one last question on the same line. You found a mutation related to resistance to medications in the chronic ones only. Do you know if those patients were actually on treatment for hepatitis B? I'm assuming so, or?
Yes.
Okay.
There are some patients which do not take the medication or no, they are not very consistent or they are not very consistent with the medication. So we don't have a lot of clinical data, but they were all treated theoretically. Yes.
Any other questions? So Belen, thank you so much again. Very nice presentation. Appreciate it. And I know you're actually in the middle of a grant review session right now. I think I understand you took a lunch break to give the presentation right now. So further appreciation for that. Okay.
Yes, thank you.
And so thank you. And then our next speaker will be Dr. Xavier Revelo. Dr. Revelo is an assistant professor in the Department of Integrative Biology and Physiology at the University of Minnesota. And Javier has been at the university working very successfully for quite a while. And we just found out about each other, I think about a month or two ago, and some of the common interests that we have. And Javier has done a tremendous work on non-alcoholic steatohepatitis in animal models. And he will talk to us today about the immune mechanisms of non-alcoholic steatohepatitis. Xavier, if you want to take the stage, it's all yours. And thank you for being here today.
Thank you for the kind introduction and the invitation to present today. I hope you can hear me okay. And so what I'll try to do is in the next 20 minutes, give you an overview of our latest work looking at the immune mechanisms that drive non-alcoholic steatohepatitis. Specifically, I'll tell you a story about B cells, B lymphocytes, and our findings that these cells can actually instigate disease progression. First, I need to acknowledge the people actually doing the work, especially PhD student Fanta Barro, who performed most of the experiments that I'm going to show you today, as well as the NIH for funding the majority of this work. The lab is located in beautiful Minneapolis, Minnesota. I can tell you right now, it's not looking like this. We have about, 5 to 6 inches of snow on the ground, and it's a cold day today. As you know, non-alcoholic fatty liver disease is a progressive condition. It begins with the simple accumulation of lipid, also known as steatosis, and through mechanisms that we truly don't really understand completely, there's a triggering of inflammation leading to steatohepatitis, fibrosis, and in extreme cases, you can have cirrhosis, and as you well know, in some cases, HCC. My lab is interested in understanding what are the factors triggering that inflammatory response, hopefully to provide mechanistic insights so that other people can find new therapeutic targets. So what we know about the inflammatory process in the NASH liver is that Kupffer cells, those tissue-resident macrophages, are central to the initiation of the inflammatory cascade. They release inflammatory cytokines, IL-6, TNF, for example, that can affect the hepatocytes. Those hepatocytes become inflamed themselves, they die, and you have this inflammatory cascade that is really detrimental for the progression of NASH. Recent work has implicated other immune cells, natural killer cells, dendritic cells, natural killer T cells. A couple years ago, when I was a postdoc, we showed that CD8 T cells drive NASH driven by type 1 interferon responses. And in some of these earlier studies, we always saw B cells present in the liver of NASH mouse and patients. And we realized that little was known about these cells that comprise about 30% of immune cells in the mouse liver at least. So we wanted to understand what is the role of these B cells, and our hypothesis was that these liver B cells can trigger inflammation and promote the transition from simple lipid accumulation to the inflammatory condition. And then we wanted to understand the mechanisms of their cell activation. We thought that changes in the gut microbiome would be the instigating factor for the expansion and activation of these B cells. If I lose you at any point in the next few minutes, you can always go and take a look at a recent publication in the August issue of Hepatology. It was featured as the issue cover, and here you can see a B cell interacting with T cells and responding to gut microbiome changes to promote NASH. So the instigating experiment that led to this study was the finding that B cells infiltrate the liver in NASH patients. We had access to specimens from people with increasing NAS scores. And what we found was that unlike seemingly healthy people with NASH had increased presence of CD19 positive cells in the liver. And so we found a strong correlation between the NAS score and the presence of B cells in the lobules and also in the portal triads. So that instigated a question: what is the role of these cells? What are they doing once they infiltrate in the NASH liver? So to study the mechanisms of B cell infiltration, we use a mouse model of disease consisting of a Western diet. We call it a high-fat, high-carbohydrate diet. We feed mice this diet for about 15 to 20 weeks. This diet has 40% of the calories coming from fat and 40% from carbohydrates, especially fructose, sucrose, and it also has 2% cholesterol. So really a McDonald's type of a diet that when you compare it to a normal chow diet, you see liver pathology here, steatosis, which is usual for a high-fat diet, but also the goal of this model was to induce fibrosis, as you can see in this trichrome staining, very strong sustained collagen deposition following 15 weeks of dietary feeding. And we've done all the metabolic testing, ALT, AST, you name it. These mice have NASH. Once we had the model established, we proceeded to study the immune landscape of these mice. And we used several technologies. The first one, we use mass cytometry, which is a technique that uses more than 35 metal conjugated antibodies that you can use at the same time to characterize the immune cells in a given tissue. So here you have the immune landscape of a lean, normal chow-fed mouse and our NASH Western diet-fed mice. There's lots of changes, as we know, especially in the monocyte, macrophage, dendritic cell compartment. But for this study, we decided to focus on the B cells, which you see here in yellow. Again, the point here is that about 25 to 30% of the cells, the immune cells in the NASH liver are B cells. So we wanted to study what is the role of those B cells. And we started by characterizing their cytokine profile. So we extracted the B cells from the livers, and we culture those cells in vivo, in vitro, with LPS, and then we assess the level of TNF and IL-6 release in the supernatant. And to be fair, we compare that with the release of neutrophils and macrophages in similar conditions. And as you can tell, macrophages make a lot of TNF and IL-6, not surprisingly, but B cells make substantial amounts of TNF and IL-6. The key data here is that in the normal chow lean mouse, the levels of these cytokines is low, but the liver environment, the NASH condition, really pushes these cells to release more TNF and more IL-6. So we began to understand that these cells are pro-inflammatory. And that was our initial take. These cells are making more pro-inflammatory cytokines. So to look at the cause effect of these cells, whether this activation that we see is actually promoting disease, we use a mouse model that lacks these B cells. These mice have a mutation on the heavy chain of the immunoglobulin M, rendering deficient in mature B cells. So these mice, these MUMT mice, lack mature B cells. However, they don't have any developmental issues at all. And in normal child diet conditions, they don't have any metabolic complications. So we fed either wild-type mice or mice deficient in B cells the diet for 15 weeks, and then we comprehensively characterize NASH progression as well as the activation status of these B cells. Long story short, because we only have a few minutes, lacking B cells in the mice protected the mice from NASH. You can see some examples here. This is trichrome staining, showing practically no fibrosis in the mice lacking B cells. An interesting finding that we're hoping to follow up on is that these mice are not protected from lipid accumulation. It seems like most of the effect is on inflammation and fibrosis. B cells not really needed for lipid accumulation. You see ALT, AST is reduced in B cell deficient mice. Oops. And we also did gene expression analysis to confirm that fibrosis, these are fibrosis genes, and these are inflammation genes, both downregulated in B cell deficient mice. So that was exciting. Then we wanted to understand more about the mechanisms behind that. What's the biology behind those B cells that make them act like pro-inflammatory cells in the NASH condition? So we use single cell RNA sequencing to better characterize the cells in the NASH liver. The strategy was to sort immune cells from the liver of normal child lean mice or NASH Western diet fed mice. These enriched CD45 immune cells were labeled with hashtag oligo antibodies, which allow you to pull and multiplex multiple samples in one tube, in one sample, and then you can send these for single cell RNA sequencing. And I'm not an expert, so I apologize if I cannot explain this correctly, but big picture, the way that single cell RNA sequencing works is that there's a barcoded gel bead that will capture one cell at a time within that bead. So as that cell passes through the instrument, the barcode will allow you to remember where the transcript and therefore the cDNA comes from at the single cell level. So at the end of the day, what you have is genetic information or transcriptomic information per cell. And doing this, we can sequence about 20 to 30,000 cells at the same time. So we did that in our mouse model, and we detected substantial changes in the immune compartment of the NASH liver, interesting changes in the macrophage monocyte compartment, and we're studying that now. There's also substantial changes in T cells, which I'm highlighting right now. But then going back to our main interest in B cells, we detected 4 different subpopulations of B cells that were changing in response to the Western diet. And here's the quantification. I understand there's a lot of data going on here, but if you focus on the changes in B cells, we see that cluster 1 remains stable. We now know that that cluster is mature B cells that resemble splenic follicular B cells, the ones that would produce an antibody in response to a virus, for example. And then surprisingly, we found 2 additional subsets that decrease with the NASH condition. We now know that cluster 11 is high in mitochondrial genes, and cluster 13 is more like an immature B cell cluster. So we're trying to understand what are the implications of these changes in B cell clusters. We went deeper into the transcriptomes of these B cells, and we found, similar to our mass cytometry experiments, that these B cells in the Western diet condition, they behave like pro-inflammatory immune cells. They make interferon gamma, they make TNF, they make interleukin-1 beta, okay? We didn't find evidence of class switching or antibody production. So these cells are behaving like innate immune cells, if you will. We did all the pathway analysis, and one of the main hits was this molecule, MyD88. And MyD88 is, MyD88 is an adaptor protein that sits upstream from inflammation and downstream from toll-like receptors. And so to determine whether MyD88 is specifically involved in the activation of B cells in this model, we created a mouse lacking MyD88 specifically in the B cells. We crossed CD19 Cre mice with MyD88 fluxed mice to make that mutation. And what we found is that mice lacking MyD88 specifically in B cells show decreased B cell activation. These are activation markers. And they also show a decrease in T cell activation. So messing with MyD88 in B cells affect T cell activation in the NASH liver. And replicating the findings from the B cell depletion model, these mice lacking MyD88 specifically in B cells are somewhat protected from liver fibrosis. Okay, now moving a little quick here because I only have 10 minutes left. We wanted to understand what is driving the activation of B cells within the NASH liver. We thought changes in the microbiome would be partially responsible. So to test that, we use a human fecal microbiota transplant model that consists of mice that are treated with antibiotics to deplete of the indigenous microbiome. And then we oral gavage microbiota preparations from human patients. We had access to 3 or 4 patients with NAFLD and NASH, so we tested both. And we compared the response of the mice to that FMT relative to a healthy, obese individual. We let the mice develop disease, and then we study disease progression as well as B cell activation in the recipient mice. So lots of data here. I'm gonna show you, or I'm gonna discuss the highlights. The main one is that transferring the microbiome from the NAFLD human patient transfer the phenotype. So these mice started to gain weight. Remember, the mice are fed a normal chow diet, same conditions, but just giving them an FMT from an NAFLD human leads to changes in body weight, a mild change in liver weight, and some increases in steatosis. Remember, it's only a few weeks following FMT. We also see some evidence of liver pathology in the NAFLD recipient mice, and more importantly, we found that the B cells in the liver of the recipient mice were activated. So we have some proof that changes in the microbiome associated with an NAFLD condition at least is partially responsible for the activation of B cells in the liver of these mice. So that's what I have to show you today regarding this study. Here's the working model that we're proposing to summarize the main findings that I've shown you. We think that in the healthy liver, B cells patrol for antigens. That's a homeostatic role of these cells in the healthy liver. However, with disease progression, specifically with the transition from simple steatosis to NASH, you have microbial antigens leaking from the gut into the liver because you have a compromised intestinal barrier at the gut level. And it seems that this translocation of bacterial antigens can activate the B cells using a MyD88 dependent pathway. The B cells sense these antigens, they overreact, they hyperactive, and they make IL-6, TNF. They induce fibrosis, and also they seem to alter CD4 and CD8 T cells. So what's next for us? Trying to understand the specific mechanisms by which the B cells lead to fibrosis. That was the strongest phenotype we detected in B cell deficient mice. So we plan to study how B cell metabolism, specifically glycolysis, is involved in the activation of these cells. And also we want to learn the effector molecules released by the B cells that activate the fibrotic pathways. Specifically, we're looking at B cell stellate crosstalk in the fibrotic liver. Some of the new advances in technology that we're hoping to use, we're planning to use spatial transcriptomics to learn more about the relationship between the transcriptome of these cells and the spatial location of these cells within the liver architecture. So we're using spatial transcriptomics. Very briefly, in the next 1 minute, we're using GeoMx, which is one of the 2 technologies available to spatial profile paraffin and BEV tissues. And the way that it works is by using probes against RNA or messenger RNA. in this case, we're using an assay that includes the entire human transcriptome. Okay, so you have more than 18,000 probes that are added to a tissue section, paraffin embed, and then using ROI selection, we decide what areas of the tissue structure we are interested in sequencing. And so through photo cleaving, those probes become cleaved from the areas of interest within the tissue, and only those probes become cDNA for sequencing using 10x genomics. So what you can do is you can use this information, the transcriptomic information from that ROI selection, and bring back that information to the histology slide and ask spatial questions. So let me show you an example, see if I can illustrate that better. We had access to NASH specimens, and also 2 samples from a NASH patient before and after vertical sleeve gastrectomy, which is a type of bariatric surgery. So the strategy we used in this preliminary study was to zonate the liver into zones 1, 2, and 3. We know there's metabolic differences between these zones, and we use CD68 as a macrophage marker, as well as CK to be able to properly zonate the 3 zones in the liver specimens. And then we use GeoMx to characterize the transcriptome of the 3 zones among these 3 samples. Okay, so there's a lot of data, we're working on that, but very nicely we see a very strong separation between our 3 conditions in the transcriptome profiling done by GeoMx, and we're looking at the specific signatures in gene expression that are changing between the before and after bariatric surgery conditions. So I have all this data, and I'm working on that. If you have specific questions, if there's any targets of interest that you have, I'm happy to look at it. Because of time, I'm going to stop here. I'm going to stop here. The last piece of data I have here is just showing that following bariatric surgery, there's a marked decrease in inflammation, specifically in zone 1. And so that's where we're headed with using spatial transcriptomics in this study. So that's all I have for you today. Thank you again for the invitation. Apologies if I went too fast. Trying to summarize everything in 25 minutes is sometimes a bit challenging, but I'm happy to take any questions you may have.
Xavier, thank you so much for a great presentation. It was very clear, very elegant. I myself have a bunch of questions, but I would like to maybe open the floor for others first, in case anybody has a question. Pablo.
Hello, everybody. Thank you very much for this moment. And thank you, Xavier, for such a great presentation. I'm excited for the technology you're showing. I was wondering about the data you presented at the beginning. Between the single cell and what you see with after doing the microbiome transplant, the single cell was showing that the B cells were going down. Is that right? And then when you do the transplant, you see the B cells going up. Is that because of the technology, or how do you discriminate? That's question 1. And the question 2 is, I see that in your model, you're proposing that the B cells are pro-inflammatory, but you didn't mention by name, but you put it in the model, and I'm wondering about it, is the galectin, the LGALs 1 and 3. Can you comment on that? Because that would be more like, instead of pro-inflammatory, more anti-inflammatory. So those two things. Thank you.
Yeah, of course. Very good questions. Let me try to answer the first one. Good catch. The detail is that in terms of frequency, the proportions of B cells don't change. You're correct. However, when you count the cells, then you can detect a substantial increase in B cells as other immune cells. So what you're seeing here is just the proportion of B cells relative to the other immune cells. That seems like it doesn't change. But overall, there's such a strong infiltration of all immune cells that if you count one cell at a time, you're going to have more B cells. And so that would be consistent with the FMT data showing an increased accumulation of cells following the FMT. So that's question one. Galectins, yeah, that's interesting. Although galectins, and you know, it's controversial what exactly they do, and it seems like galectin 1 could be a little different from galectin 3. What we're seeing is that there may be a separation between pro-fibrotic and pro-inflammatory effects of these galectins in the liver. And so we're trying to understand that better. And at this point, it's inconclusive. We're doing co-culture experiments between B cells, hepatic stellate cells, macrophages, trying to dissect that complexity, because you're correct. It gets a little complicated. What's pro-fibrotic, what's anti-inflammatory? It's a little tricky. Thank you.
Jesus. Sorry, Jesus.
Yes, thank you very much. Congratulations, very, very nice study, very complete. My question is, did you, maybe I missed it, but did you analyze potential autoantibodies developed by these mice?
Yeah, great question. Not specifically, but we are looking at the idea of the B cell receptor becoming engaged. And we're using mice that have a transgenic B cell receptor and seeing if that is also involved in B cell activation. I suspect it's going to be very hard to find detectable autoantibody levels in the circulation. So to be honest, we haven't tried. And we see that most of the transcriptomic data points at a toll-like receptor activation, not a B cell receptor adaptive type of response. So we haven't properly looked at that, but at least in terms of class switching, we're looking at whether class switching is involved.
Perfect, thank you.
Yep, thank you.
Thank you. Marco?
Thanks. I just would like to congratulate Javier for your really nice work, top-notch research indeed. Some of the concepts of adaptive immunity are in NASH are exploiting now. I mean, all the focus was in innate immunity, and now adaptive immunity is. And I would like to ask a couple of questions. They're very basic though. One is there any data about B cells depletive agents like rituximab in these models or in liver disease other than in autoimmune disease? And second, which are your thoughts, because you just showed the picture of the thesis, but about the role of B cells in liver fibrosis? I mean, that is known long ago, but now with all your technologies, you are really investigating the crosstalk between B cells and hepatic stellate cells, likely, which is, as another situations, bidirectional. But what is known about this specific interaction? And the last question, I'm sorry about this, is I got puzzled by the MID88, which in macrophages is very important in the activation of inflammasome. And I wonder if the B cells also have that as a mechanism. Thank you. I'm sorry for this, for so many questions.
Oh, no worries. I'll try to remember all three. So rituximab, no human data, no clinical data to showing any or to investigate the role of depleting B cells in NASH or NAFLD or any of the related conditions. We've used it in mice as an alternative strategy to the genetic model, and it works well depleting B cells. In mice, the depletion lasted the 15 weeks of the dietary treatment, and that replicated the effects of the genetic depletion. Then it'll be, so someone probably needs to study that. I don't know if that idea is going to be well received because of potential immunocompromising effects of B cell depletion strategies to treat a chronic condition. Instead, I feel like if we understand the mechanisms behind activating B cells, maybe we can more precisely target some of those pathways. So that's my take. The third question about inflammasome, we don't know. We don't see a signature of inflammasome activation by RNA-seq. It doesn't mean it's not there, but you're correct, MyD88 sits upstream from NLRP3 as well as NF-kappa B. Which one is here and clear? And I'm sorry, I'm forgetting your second question.
That was the one about a little bit more of detail on the role of B cells in fibrosis.
Yeah, so there's a couple
hypotheses.
Yeah, a couple of studies showing that B cells are involved in acute models of liver fibrosis, carbon tetrachloride injected mice, no B cells there, those mice are protected. And so there's some premise for what we see in the NASH model. The difference clearly is that an acute model of liver fibrosis is not the same as NASH because you have the metabolic complications associated with NASH. So it seems that this profibrotic role is consistent, but I wouldn't argue that is a similar process by no means.
And do you know of how is the potential interaction between B cells and hepatic stellate cells?
Yeah,
what is known about that?
Yeah, we're thinking about it. Two options. The first one is secreted factors, so cytokines, chemokines that the B cells would secrete. The other one is direct interaction between B cells and hepatic stellate cells. And so we're using in vitro systems to test which one is potential in both, and then we'll move to a mouse in vivo model. So I see that as the two potential ways: direct contact or secreted factors.
Yeah, extracellular vesicles might also have a role there.
Yeah, that's a good idea.
Thank you again. Really fantastic presentation, and congratulations for your nice work.
Thank you, Marco.
Xavier, thank you so much. I have 3 questions, but I won't say any word, otherwise we're not going to ever leave this meeting. But that was phenomenal. So thank you so much, both Belen and Xavier, for presenting today. So for the Escalon seminar proper, I think we're done. I think we have a general assembly meeting. What I'll do is those that need to stay for the general assembly, I'll give you a 2-minute, that's okay, break. So in case you need to go to the bathroom, take a glass of water, just please 2 minutes, and then we'll see you back here. And for the other ones, thank you very much and have a nice day.